Sister Nivedita University, we extend our warm welcome to everyone for being present here for the seminar on genetics and plant breeding in agriculture. Today, we are honored and we feel privileged and welcome our honorable chief guest of the seminar, Dr. Shubhendu Chatterjee, former Director ex officio, Department of Agriculture, West Bengal. Now, I would request our honorable chief guest and our director sir to come over to the stage for watering of plant. Thank you, sir. Now, I would request our respected director, sir, to kindly introduce our chief guest to the member present here.
Good morning to you all. All you are aware that the School of Agricultural Science of Sister Nivedita University Good morning to you all. You are aware, my dear students, you are aware that today the School of Agricultural Sciences, Sister Nivedita University, has organized this valuable, important seminar on topics, a tale of two genes that revolutionized Indian food production or food production in India. Very important in the subject of generation plant breeding, you are reading already in your curriculum. The course is there, generation plant breeding. Within the generation plant breeding, you are aware that several disciplines like crop physiology, seed production technology, genetics, plant breeding, all the subjects are in the generation plant breeding group. And first year to fourth year, you have to carry out several courses from this discipline. So we took this opportunity to invite that our ex-director, former director and ex-officio secretary of Government of West Bengal, Department of Agriculture, Dr. Shubhendu Chatterjee as guest of honor or chief guest, to address these topics which will be benefited, you will be directly benefited. You know, Dr. Shubhendu Chatterjee is a very renowned scientist and he was in the research wing of Government of West Bengal for several years. From Pulse and Wild Seed Research Station, West Bengal, that is at Bahadurpur. Then the Rice Research Institute at Chuchura, all the director, everywhere he was director of that station. Then finally he come to Director of Agriculture over the state and retired from there. Still, she is very much active. Whenever we are calling him, he is coming and we are encouraging us and giving us impetus. So on this occasion, I am happy to welcome Dr. Shubhendu Chatterjee in this session to address the student. It is the rare opportunity you will be getting. So you please, if listen minutely and sincerely, and whatever you have any question, there will be a brief session on question answer, and you will ask the question, he will answer you. Then I am welcoming also the students that this type of seminar we are organizing first from the School of Agricultural Science. You know we have a shortage of seminar hall. So we are doing all in the department or agriculture faculty. That is a small room, and we are not accommodating you all in any function or any type of conference or meeting altogether. It is almost impossible. So we have got this hall for 2 hours only, because it is also only hall, seminar hall, the only hall of this university now. So large and well equipped. So we shall vacate it, but utilize this time. With these few words, I am inviting Dr. Subindu Dev Chatterjee to address the student and go to the talk this time. No problem. Okay, thank you very much.
Director School of Agriculture Science, the learned teachers of this institute, and the students. The most important part of this educational institute. At the very outset, I like to convey my deep sense of gratitude for being invited in this esteemed institute for delivering a talk. As the institute is named after the Honorable Soul's Sister Nivedita, who besides her missionary work was pioneer for expansion of education, where he had she established 2 schools. She started his career as a teacher at the age of 17. And as she attained the mastery in as educator, she started to establish schools. First, she started establish school in London. In vogue in most of the nursery schools. She came to India in 1898 as a disciple of Swami Vivekananda, and in 1898 Vivekananda initiated her the vow of Brahmacharya. And in 1899, and in 1899, she developed and established a girls' school at Bagwaya. So, throughout his life, she was a champion for expansion and spread of education. And it is quite natural that an university established after his name will offer education in agriculture science, which is basically devoted for the downtrodden farmers of the society and of this country. Now, before I go to today's topic, I like to say a few words to the students. I think many of you are still having a great dilemma. Why to study agriculture? Particularly in a situation when your friends are madly running after computer science, different branches of information technology, engineering, etc., etc. Now, agricultural science combines many subjects: biology, chemistry, engineering, and it offers a diverse and interdisciplinary education. It is an innovative sector and basically concerned with the recent innovation of the science in agriculture use. I can give you two glaring examples. Now you know, in the field of molecular biology, the great invention is that the genes, you know the genes are responsible for all the characters. Now these genes are not marked for any organization, but it can move from one organization to other, just like musical notes, sa re ga ma pa dha ni sa. Now the agricultural scientists, utilizing this invention, what they did, they transferred the cry-AC and cry-AB gene. You have heard the name of cry-AC and cry-AB gene? You heard? You don't have heard. So they have transferred the cry-AB and cry-AC gene from a soil bacteria named Bacillus thuringiensis. Now this cry-AC and AB genes in the soil bacteria is responsible for develop a insecticidal protein. What is insecticidal protein? That is a protein that kills insect. A protein that kills insect. Now they transferred these two genes to cotton plants to develop cotton variety that can protect cotton bollworm, which is a devastating paste for the cotton farmer. And in this way, the BT cotton has come. And now, if you look, the BT cotton, 75% of the cotton area is under BT cotton. So the agriculture technologists, the agriculture scientists did this job for the downtrodden cotton farmer. Now again, I think many of you, including the teachers, are hearing this gene editing word. I think it is now very popular also in the newspaper, in the media, in the electronic, as also the print media, the gene editing. Now what is gene editing? Gene editing is alteration of genetic material in living organism, that is deletion, duplication, or replacement of a gene sequence. You know the gene sequence, this DNA sequence, the ATGC sequence, you know. Gene editing means the changing of the sequence of a particular gene. And by changing that sequence, you can altogether change the functioning of the gene, that is, change the production of the protein. Now this particular innovation has been utilized also in agriculture by the scientists of IARI. They have edited the gene of basmati rice. You used to eat basmati chal. So I think you know the basmati varieties. It is specially grown in the North Indian condition. The North Indian farmers are basically the basmati farmers. Now their main problem is the weed problem. Now they don't get this much of water that we get in Bengal. Their rice cultivation is somewhat dry cultivation, and they face huge problem of weed. But they cannot spray the herbicide to control weed, and manual weeding is very costly, and they have to incur huge expenditure for manual weeding. So they appeal to the scientists, North Indian scientists, to do something for them. So the IRA scientists, they have changed the gene sequence of ALS gene. That is one gene. Now you know the herbicide, I think the fourth year students have read it. The herbicide are, for paddy it is mostly it is basically pre-emergence. There is no post-emergence herbicide for paddy. Now, the indiscriminate herbicide like imjatifer, it kills the paddy plant by binding the site of the amino acid production. That is, it retards the growth and kills the plant. Now, by editing the basmati, this ALS gene, this binding site they have deleted. And now, this gene edited basmati 1979, they can safely spray this indiscriminate herbicide and can save huge money they used to spend every year for the control of the weed. Don't you feel these are very innovative activities in agricultural science or agricultural transfer of technology division, which we will have to do after pass out. Now, beside this, the agriculture is the main driver and the crucial for our food security. Now, in 2050, the government estimated that we will be needing 333 million tons of food to maintain our food self-sufficiency, as against the current level of production is 225 million tons. Now, with hard toil, the India was initially a famine-stricken, chronically food deficit country. Now, it became self-sufficient in food only in 1980s. Until date, India is self-sufficient. Till date, India is running self-sufficient. And you, after pass out, are to hold this button to keep our country self-sufficient all along. Besides this, if you think of the job potentiality, everybody is interested for their job after pass out. Now see, agriculture is the biggest private entrepreneur in India. It is the biggest private entrepreneur. Now 50% of the people sustenance is coming from the agriculture. So it is the biggest. So there are enormous job potential in the government sector, in the private sector, in the say in the fertilizer industry, in the insecticidal industry, in the financial industries like bank, in the government's extension officer, in the NGOs, those who are working for the farmers. There are huge and enormous potentialities of service. So I think you have chosen the right subject, and it will help you to build up your career. I have a list of service sectors, but I don't want to read it here. I think you will be getting it from your institute. Now with your permission, I am going straight to the topic. Now today's topic is a tale of two genes that brought revolution in food production in India. Now see, when British left India in 1947, our food situation was really grave. Now at that time, India suffered, continued to suffer and haunted. You may not have heard the famine of 1942-43 of Bengal that took away millions of lives only for failure of rice. Now again, after partition, the partition took away the major wheat area of Punjab, and the partition took away the major rice area also in East Pakistan at that time, and now it is Bangladesh. In addition, partition landed with millions of refugees loaded in West Bengal region. Now the population census in 1950 projected that at that time the population of India was 361 million and the food production was only 50 million tons. So, and that was too poor to meet 2 square meals a day for the people. And again, the scope of import of food from other countries or other sources became very scary in a world just recovering from the World War. Now, after great difficulty, India could make USA agreed to supply food under a special PL 480 scheme, where the payment, where the India can make the payment for the supply of food in rupees rather than dollar. As because at that time, the dollar reserve of our country was too poor to make any payment for the food. So they had to agree in this proposal. So the agreement was made in 1954. And the first consignment of food came to India in 1956, and it continued up to 1959, and that in that period only 10.68 million tons of food could be supplied to India, and the majority of that was wheat. But up to this period, in agriculture sector, there has been no significant growth in production front. Basically, India tried to expand or increase production by increasing area. That is, area increase approach was taken initially, but that did not pay. At this time, the government and the policymaker realized that country cannot wait further to go on begging for food. Although they were compelled to renew this PL 480 agreement for a further period up to 1964. And they asked the scientists of ICAR, IARI, and other research station. At that time, most of the agriculture research was under the State Department of Agriculture to develop seeds of our principal food crops. And our principal food crops in India is two. One is wheat and the other is rice. Accordingly, the Indian scientists and all the agriculture personnel concentrated their efforts to invigorate the seed, genetically invigorate the seed to yield high. As because at time the yield potential of the varieties under cultivation, both under cultivation, was only 522 kg per hectare. Now, FAO came forward at that time to help India to transform the rice variety to yield more. So they started Indica japonica hybridization program, as because Indian or Indica rice were tall, prone to lodging, low yielding, low tillering, etc., etc. As against this, the japonica rice, that the rice that are grown in Japan, are short stature, steep straw, and non-lodging type. It was thought that if the japonica blood could be incorporated in indica rice, then we could get a stiff straw, non-lodging rice variety that could yield much more than the present. But the attempt also failed. So by this way, we spent many years. It is 1964 and we became free India in 1947. So 17 years in this hunt for high yielding rice and wheat. Now, report came that in USA and in Philippines, they are exploiting two genes. They have exploited two genes and got very high yielding wheat and high yielding gene, entirely transforming the idiotype of the plant of both wheat and rice. So India administratively decided to export those two genes to India. And these two genes are RHT gene in wheat and HD1 gene in rice. The first we see the role of RHT gene. The miracle of RHT gene in transforming the architect of the wheat plant. The normal wheat plant was taller than 150 centimeter. RHT gene reduced the plant height to 60 to 110 centimeter. As a result, the traditional wheat plant underwent revolutionary morphological and physiological change. Morphological change is reduced plant height, A reduction in plant height improved lodging resistance, which was a serious problem in wheat cultivation when high rates of nitrogen fertilizer were applied. If you want to get more yield, you need to feed the plant with more nutrient. Consequently, from very early in the developmental stage with reduced plant height or RHT gene, there is a reduced competition by the stem for the nutrients, leading to a greater portion of dry mass being partitioned to the RHT genotype ears. Since length of the stem has got reduced, so nutrient expense for production for elongating the plant height is minimized, and as a result, this savings in the dry mass, that is, this partitioning or this saving results in greater number of competent floret, which also have a greater mass per carpel at anthesis. Carpel means your spikelets. Both these features favor increased gain set and greater harvest index than tall wheat at maturity. Harvest index is nothing but how much of biomass is being diverted to the grain or to the kernel. If it is more than 50%, then your yield will be very high. Both these features favor increased grain set and greater harvest index than tall wheat at maturity. Greater flow of dry matter ultimately allowed the ear head to grow larger, bear more grain per spike, and finally yield higher. Two mutant alleles, RHT-B1B and RHT-D1B, discovered Norin 10 wheat. The vessel through which the gene came to India is Norin 10, and that was a variety of Japan, and this variety brought about these changes. Now the journey of the RHT gene. These two genes, these two dwarfing genes, were derived from Norin 10, which was semi-dwarf variety bred in Japan by Inazuka in 1935. So it was a very old variety. The term Norin is an acronym of the Japanese Agricultural Research Station in Latin. The ancestry of Norin is a cross between Daruma, a local Japanese wheat, with one soft and one hard wheat of USA. And this Norin 10 began to attract the United States scientists, particularly Salmon, the great wheat breeder, who bring the Norin 10 seeds to United States. And Vogel at Washington University utilized this Norin 10 seed and transferred this gene to the winter wheat variety of USA and got the very high yielding variety gains in 1940. So the grains is the first high-yielding variety, semi-dwarf high-yielding variety, but that was winter wheat. And we grow here spring wheat. So this gene is required to be transferred to spring wheat. Now this job was done by Norman Borlaug in Simit. When Norman Borlaug started to working in Simit, International Wheat Research Station, wheat and maize research station, his objective was to develop a shorter rust resistant wheat variety. So he screened 10,000 germplasm of US, but did not find a single genotype of this type. So he requested Vogel to send the Norrington genes to him. She got the Norrington. This is the Norrington derivative. You see the plant height. Norrington, actually it was a lucky break providing short stature as also rust resistance. Rust was a problem of the wheat in Indian varieties. After a series of crosses and recrosses, Norrington with Mexican wheat varieties, the Borlaug developed a new type of spring wheat, short and steep stalked varieties that tilled profusely, produced more grains per head and were less likely to lodge. By 1962, from this series, two varieties, Pictic 62 and Penjamo. Penjamo, I think, is known to some of you. And from this series, this Sonara 64, Sonara 63, Larma Rojo 64 came. I think now these names are coming to your mind, Professor Jaman. Sonara 63, Sonara 64, and Larma Rojo. Now this semi-dwarf wheat progeny was so high yielding that India also got this news. Now see the genesis of this variety, how it was evolved by the breeder, what type of different process was made. Now the mechanism, how the gene make the tall plant so dwarf. What is the biochemical pathway? Actually, the reduction in stem elongation in varieties containing this allele is caused by limited response to a phytohormone called gibberellin. Now, gibberellin elongates the height. RHTB1, RHTD1 encode DELA protein. DELA protein, all enzyme is to produce a protein. And this protein repress GA responsive growth. They are mutant alleles confer dwarfism by producing more active forms of this growth repressor. This semi-dwarf allele introduce premature stop codon. Stop codon means there will be no further amino acid production. And this stop codon was placed in the GA20 oxidase synthesis. That is, GA20 is the ultimate product of gibberellin biosynthesis to give the plant impetus to grow longer. Now, GDDB, geranyl diphosphate, is a common precursor for gibberellins. biosynthesis of gibberellin has got 3 steps. The first step is the biosynthesis of N-curine in the proplastids. You know, the students, what is proplastids? A small colorless organelle that give rise to plastid. Pro to plastid, precursor to plastids. Then conversion of N-curine to GA12, and then formation of carbon-20 gibberellic acid and carbon-19 carbon-12 GA12 and GA53 and carbon-19 GA4 and GA1 in the cytoplasm. In the third step, from GA53, GA12 and GA53, the biochemical pathway run parallelly, and in one pathway GA20 oxidase is produced, and if this pathway is stopped, then GA53 the quantum is elevated. So if GA53 quantum is elevated and GA20 oxidase is reduced, then the pan gets dwarf. Now impaired GA20 oxidase activity results reduced amount of GA20 oxidase causing repression in plant height. Now how this gene came to India? Now the government of India, particularly the Ministry of Agriculture, requested Dr. Borlaug to visit India. Dr. Borlaug came to IARI and visited the wheat genotypes of the country and decided to send a series of his material to India. And this was in 1963. Among these, Sonara 64, Mao 64, Sonara 63 and Larmarojo 64 were sent in bulk quantities for conducting multi-location trial. Now in multi-locational trial, our scientists find that Sonara 63, Sonara 64 and Larmarojo 64A on an average registered 35 to 45% higher yield over best strain. Union Minister for Food and Agriculture took the decision to import 18,000 tons of seeds of Larmarojo and Sonara 64 from Mexico. The seeds arrived in September 66 and were planted in 240,000 hectares in the year 1966-67. Between 65, 66 and 67, 68, India's wheat production rose from 10.4 million tons to over 16.5 million tons, crossing 20 million tons in next 2 years. See the jump only by exporting these genes through these 3 vessels. Now the miracle change, you see the change, this is the graph. This is the wheat yield. This is the wheat yield. And this is the wheat area. You see the change. So ultimately, the begging bowl of wheat gradually became filled up with country's own production following spread of RST in this variety. So this was a job of the agriculture technologist and what you are going to be. Now come to rice. You know, rice is the most cultivated grain in India, where over 50% of its population depends on its sustenance. The single most important staple item in almost all India's extremely diverse cuisine. It is so important that a failure of rice crop in 1942-43 brought about the Bengal famine and millions of people died. Shortly after independence in early 1950s, when India was thriving for food, rice covering the largest area, but contributing only 41% to the Indian food bowl. And the primary reason for this poor contribution was low yield. The yield was only 0.6 ton per hectare. That is 600 kg per hectare. The cultivated varieties were tall, more than 150 centimeter, weak stemmed, extremely prone to lodging and low tillering. Lion's share of the biomass produced by such types was utilized for vegetative growth, leaving less to produce kernel. So the plant type need to be changed. India's effort to develop steep straw reduced height rice variety started in 1949, which I told you earlier, the FAO-sponsored Indica japonica cross. This project developed two varieties. One is ADT 27 and the other is Masuri. I think the Masuri's name is popular to some of us present here. But they were well, but not fit for the quantum jump which wanted. Then the Ford and Rockefeller Foundation established in 1963 the International Rice Research Station at Philippines with the objective that they are to produce first the short stature, non-lodging, high-yielding rice variety for the half of the world population, which are solely dependent on rice for their staple foods. Now the IRIS team recognized that rice had a problem of lodging when heavy heads were supported by tall, spindly stalk. So they imported seeds they collected 10,000 seeds from all over the country. Now, Dr. Peter Jennings in 1962, from this 10,000 germplasm, he selected some parents and made 38 crosses, only 38 crosses. And out of the 38 crosses, one cross number 8, was a cross between a tiny short stature variety DGO Ujen. DGO Ujen means short leg. It's a Japanese variety. Taiwanese variety. DGO Ujen means short leg, pink tip, with a Indonesian tall variety Peta. And he got only 130 cross seeds. And this 130 cross seeds he grew in the pots. And from that pot, he got a good amount of F2 seeds. And those F2 seeds which were grown in the plot, and of this F2 seat, the F1 seat, one interesting fact was that all the F1 seats were tall. There was no dwarf plant. And in the F2 seat, 1/4 of the plant was dwarf and the 3/4 was tall. So he discarded the tall plants and selected only the 1/4 short stature plant. And thereby, after selection and selection and selection, in the F5 generation, he selected one plant and he named it IR8-288-3. To him, it was the most promising. And from this selection, IR8 came. This new rise progenies were 110 to 120 centimeter as against 150 centimeters, so height was reduced significantly. Have strong stem that held huge head up when heavily fertilized as compared to weak stemmed existing rice that lost under high fertility. In 1966, a young Iri agronomist S.K. Dedatta, who was then living in Jadavpur, I met him at Calcutta University. He also was an agronomist. He actually identified the IR8 and showed by his experiment that this particular variety can yield 10 tons per hectare at 120 kg nitrogen and can yield 5 tons per hectare under 0 kg nitrogen. And it matures in 130 days as opposed to 160 to 170 days for traditional variety. Now see, the peta is in the middle. On your left is IR8 and on your right is DGO. So from that was tall and this is semi-dwarf and that was the dwarf, short stature. So it is in between. Now the gene behind this dwarfing and the transformation is HD1 gene. And the gene is located in chromosome number 1. Rice has got 12 chromosomes, 2n is equal to 24. It is one of the most important genes deployed in modern rice. This gene in recessive condition, you know dominant and recessive condition, I think. It is in recessive form, result in shortened calm with improved lodging resistance and a greater harvest index, allowing for the increased use of nitrogen fertilizer. And the donor of the gene was DGO gene, DGWG. The HD1 gene encodes, again here also the HD1 gene encodes gibberellin 20 oxidase, GA20 oxidase. Now, and this effect is also similar. This biochemical pathway is more or less similar what we discussed in the event of wheat. This impaired GA20 oxidase here also reduce the plant height. And if you spray gibberellin in the semi-dwarf variety, it will attain the same height of the peta, from which it came down to semi-dwarf type. This reduction in active gibberellin result in shorter internode. I think I can skip this slide. Now see, this is the figure. The inactivation is here by the gene. It is not working, but it is working. The inactivation is here. First stage, second stage, and the third stage is inactivated. So elevated content of GA53. You see here is GA53. And GA20 oxidase is reduced. Now, miracle in rice font. IRA was straightway introduced in farmers' field. No multi-locational trial conducted by any of the scientists of the country. It was straightway, which went to the farmers' field, large-scale farmer skill testing at Andhra Pradesh. And his spectacular performance drew the attention of the all the reporters of the country. And then gradually the IRA moved towards other southern states like Tamil Nadu and also to the rest of the country. And with the spread of this variety like wildfire, the panorama of domestic rice production started changing. 1950-51, that is just after our freedom, the country produced 20 million tons of rice from an area of 30 million hectares. In 1969-70, production rose to 40 million tons, and by the turn of the century, it touched nearly 90 million tons. That is 4.3 times increase in production, primarily for almost 3 times enhancement in yield rate. Yield rate from 668 kg to 1986 kg per hectare. Now see the reflection in country's food production by the effect of these two genes in two crops. Now domestic food grain production at the time of independence was far short of for our minimum requirement. You see in 1950, the food grain production of 50.8 lakh ton, and we got only 50.8 million ton, and we got only 4.8 million tons food through PL480 scheme. Now in 1960, it is 82 million tons, 32 million ton up, and we got 10.4 million tons from that PL480. But after this period, in 1970, you see the change. 108 million ton production, and the food grain import drastically came down to 7.5 ton. In 1980, 129, export came down to 0.8 million ton. In 1990, 176 million ton, export came down to 0.3 million ton. And in 2000, no export. And the production rose to 201 million ton. And see, the buffer stock. There was no buffer stock, and now the buffer stock in 2000 is 40 million tons buffer stock. And in 1950, our production was only 50 million tons. And in 2000, our buffer stock came to 40 million tons. Yes, population has grown. Population has grown. In 1950, it was 361 million ton, 361 million, and 2000 it is 1,000 million. Now it is 140 crore. Still, with the burgeoning population, we are still self-reliant in food. And that is due to these two genes initially exported by the Indian government. Thanks to the decision. Thank you.
Thanks to Dr. H.D. Chatterjee for his thought-provoking talk on tale of two genes that revolutionized food production of India. Actually, if the student will be directly benefited with this topic, because I told previously, all the academic calendar, over the academic calendar, you can understand that we had several course of genetics and plant breeding subject. That's why you will be benefited, I hope. If you have any question or if you have noted anything that to be discussed that you have not gone through, you may, few points you may raise, you come here, and please, one or two question you can discuss with the speaker, because his presence you will not get ever. So, sometime we shall call him also, no problem, but if you spot question you have, anything, you have noted, any faculty or any audience, you have anything to discuss or to raise the interaction with speaker, please raise, you come forward and ask the question so that a brief discussion can be, we can took some time for discussion or interaction. Please, the seminar is open for discussion.
Anybody? Madam, you want to say
Please.
something?
Please raise your hand.
No, you come forward.
At first, tell your name. Huh?
মাই নেম ইজ সুমন দাস আই এম ফোর্থ ইয়ার স্টুডেন্ট। আমার কোয়েশ্চেন হচ্ছে স্যার আপনি হিস্ট্রিটা বললেন, এবার যে ইমপ্রুভমেন্টটা হয়ে গেছে ইল্ডের সেটা হয়ে গেছে, বাট ইল্ড তো একটা কোয়ান্টিটেটিভ ক্যারেক্টার। এবার আর কি ওর কোনো ইমপ্রুভমেন্ট দরকার? মানে ধরুন ইল্ড একটা বাড়ানো হয়েছে জিনের দ্বারা, এবার নতুন কোনো জিন কি আসতে পারে যেটা আরও ইল্ড বাড়তে পারে? কারণ এখন যে ইল্ডটা বাড়ছে ওটা ধরুন ডিজিজ রেজিস্ট্যান্ট বা পেস্ট রেজিস্ট্যান্ট ভ্যারাইটি বা ধরুন বেশি পরিমাণে ফার্টিলাইজার দেওয়ার জন্য বাড়ছে, বাট জেনেটিক্যালি কোনো ইমপ্রুভমেন্ট নেই, তো ওইটার কি কোনো স্কোপ আছে?
ওকে, ঠিক আছে,
you,
বলছি। তোমার নাম? হ্যাঁ? সুমন দাস। আচ্ছা, সুমনের পরে আর কেউ কিছু?
Dr. B.C. Ghosh. Dr. B.C. Ghosh was a former professor of IIT Kharagpur in the discipline of agronomy or in agriculture. Already he is working till now after retirement. He has some question. I welcome.
Not question. I want to share some of my experience with Dr. Chatterjee. In fact, very shortly I can say, after independence, the most and best achievement of the country is this 2G. As he already told, in 1950s, our production was at a very low level. We used to import food grains. Now, by this time, we have a buffer stock extra. The whole achievement is behind this 2G is behind it. I was a scientist in CRRI Kotak. I work on rice intensively in CRRI as well as in IIT Kharagpur. So CRRI, I work on deep water rice. The rice which will be with the rise of water level in low-lying area, the height of the plant also will increase even by 2 to 3 meter tall. So a lot of fertilizer trial, etc. we did in CRRI Kotak. We have a lot of information on that. Now, thing is that some of the negative, any prosperity cannot be 100%, So there may be some negative point. That's what I want to discuss with Dr. Chatterjee, that green revolution you have heard this word, which has started from 1960s, 70s onwards. Green revolution, we have became self-sufficiency in food grain production. Population is increased. Today we are the highest population in the world. And we are feeding. That is the best achievement. Otherwise, what would be our situation. Now, on the other hand, some of the things are happening. Say, I am now working on organic farming. How to grow the crop organically. Now, Dr. Chatterjee, one line of thinking, now another line of thinking will come, that due to introduction of high-yielding variety, requirement of fertilizer and chemical pesticide also increased, and that has killed our ecosystem. So in our management, now we have to think like this. In our whole agriculture management, we have to compromise the genetical change and makeup and also sustainability of agriculture. Our agriculture now at a little difficult situation in the sense that we are using chemical pesticide and fertilizer and disturbing the biodiversity of the soil ecosystem, environmental problem. We are applying urea in the field, the nitrate is leaching down, water is getting polluted. We are spraying the pesticide, killing the useful microbes. So that also should not be allowed further. So we have to compromise organic farming and chemical farming in such a way that our soil health is sustained. Otherwise we'll face like food crisis. Food we are eating, but that also a lot of residue is coming in the food, So organic food, I used to teach a course in IIT Kharagpur, organic food chain management. So organic food production, how to do, I am doing some still in the villages working, say how to develop an organic village. The habit of using green manuring and organic fertilizer, farmers have forgotten almost. Earlier, dhoncha cultivation was very common, now it is not there. So only farmers are habituated in applying chemical and forgetting organic. So this aspect, you are the future of agriculture development. You have to work a lot we have seen one stage of progress, you have to see you have to face another phase of progress in agriculture. So start thinking how to save soil, water, environment from the agricultural activity. Thank you very much.
Okay, thank you, Dr. B.C. Ghosh, for his kind intervention and his observation on organic farming and the chemical farming. It is our in topics also. We are in a classroom. We are bolbe. Any student from you, please?
Please do come.
Put your point of interaction which can be discussed and other fellow students can learn something.
Good afternoon, sir. I'm a student of the 4th year of basic agriculture. I'm Titli Datta. So my question is,
What is your name?
my name is Titli Datta. Titli Datta. Yes, sir. So my question is that before releasing of a variety, certain trials are done, like the preliminary yield trial that is done in the farmer's field, and the multi-location trial is done in different locations. But the variety might work in that environment only better, right? And might be environmental factor that the variety is working, and so like the yield, it is passing for yield. Higher yielder, that might be working for that location only, that environmental factors only. How do you reduce this environmental effect and in producing of a new variety?
Thank you.
Thank you, sir.
Okay, anybody? Any students?
আচ্ছা এটা উত্তরটা দিয়ে নিই
আচ্ছা,
আচ্ছা এটা
okay, okay.
First, I will answer these three questions or three points raised, and then you also think what will be your question. Now, Shumon and Dr. Ghosh both emphasized one point, that is, what after the yield ceiling set by IR8 could not be overcome till date. It is true, 100% true. So both of you want more yield. Now now see, agriculture is an interdisciplinary, means in agriculture there are several inputs. Now chemical fertilizer or organic fertilizer, it may be pesticide, it may be management, it may be etc., etc., etc. But which one gives yield? Which one gives yield? Whose potential you find in the field? Is it the potential of the fertilizer? Is it the potential of the pesticides? Is it the potential of your management? No. Subijam sukhetre jayate sampodayate. Good seed, if put in the good soil, yield bountifully. If seed has got no potential, then how you will get yield, good yield? So you ought to genetically invigorate the seed to produce more yield. So whether you will go in for chemical agriculture or organic agriculture or intermingled, your ICR has going to recommended organic agriculture will reduce the yield. They have completed their all India trial. and they are going to recommend today or tomorrow that it is an organic plus chemical, say the 40% chemical, 60% organic. So you manage your soil and you harvest good. That is one part. So if sun is the only source of all energy in the universe, the seed is only the source of energy in the agri world. If seed has got no power to yield, no amount of fertilizer, whether organic or inorganic, it won't do. So the first and foremost thing is the seed. So who is reading genetics and plant breeding? It is actually you are the person who are responsible to invigorate the seed, and others are only help to express the entire potentiality that is hidden in the seed. The potentiality of yield that is hidden in the seed will be expressed only with the help of, if there is a stone in this, in a dark room, your light can help to identify that stone. But if there is no stone, your light will simply fall and it is nothing there. So you have to keep your stone, your diamond, your gold is your yield potential. And the Shumon's question is, what further? Yes, and that will be big. Now there are many attempts are being taken. One is hybrid rice, you know. You know hybrid rice? China has extensively expanding hybrid rice. Where is Sumon? Raise your hand. You know hybrid rice? It is also one attempt that rather than growing inbred, these varieties are all inbred. If you grow hybrid, the hybrid will display a heterotic effect, and it will put you 15 to 20% higher yield than any of the inbred. Say this the hybridity you have found in maize, in a vegetable, in millets like bajra, jowar, etc., etc. So hybrids are also being tested. Now the problem of hybrid in wheat and rice is this, that it is a self-pollinated crop. So production of seed is costly than that of your maize and your millets. And the second thing, the jump is not that, the yield jump is not that that high to allure the farmers to purchase and grow it. So hybrid rice is being tested. And third thing is you read QTLs? You read, Sumon? You read QTL in your classes? বলো না, আমি শুনতে পাচ্ছি। আরে বলো, আমি শুনতে পাচ্ছি। So you know that some QTLs have been identified.
আস্তে আস্তে
some QTLs have been identified that have good contribution for yield. These are called yield genes, and these genes are being tried to be pyramiding in a single variety. So this is the future. Sure. Why problem? Why problem? What problem? তুমি কি জানো প্রতি বছর তুমি যে ইনব্রেড মানে এমনি ভ্যারাইটি যে ধান তুমি চাষ করো তার কতটা হচ্ছে সেফ সিড আর কতটা পারচেস সিড তুমি জানো? তো সেটা না জানো তো সেটাও তো কিনেই করছে, তো এটাও কিনেই করবে। হ্যাঁ হ্যাঁ না করতে পারবে না তো। আরে সেকেন্ড ইয়ারে তো ওটা F2 হয়ে যাবে, হবে না তো, ওটা প্রতি বছরই হাই।
ঠিক
না না দ্যাট ইজ ট্রু, করতে হবে তোমাকে বেশি মেজে তুমি করছো না,
বেশি প্রোডাকশন পেতে গেলে করতে হবে।
ভুট্টাতে তুমি করছো তো, ভেজিটেবলে তুমি করছো তো, তুমি ফ্লাওয়ারে করছো না, গাঁদা ফুলে করছো না। মার্জিনাল ফার্মার শুধু ধানের ক্ষেত্রে না মার্জিনাল ফার্মার শুধু ধানের ক্ষেত্রে ভাই। নেক্সট হচ্ছে তুমি যে প্রশ্নটা করলে মাল্টি লোকেশনাল ট্রায়াল বলে আমাদের অল ইন্ডিয়া কোঅর্ডিনেটেড প্রজেক্ট আছে ফর ইচ অ্যান্ড এভরি ক্রপ। Say for rice, there is a all India coordinated project, and this project is run in every state, in every universities. So when you enter a variety for testing, this variety will be tested throughout the country, in all the states. So then the performance of the variety will be pooled. First, it will be pooled in all India basis, then it will be pooled in a zonal basis. Some variety will perform good in the zonal basis, and few varieties will perform good on the all India basis. So all the environments are being tested before release of a variety. Farmers' field tests are not always done. Any more question?
Any student put any question now? Please come forward quickly.
Sir, myself Pradeep Ghosh. I am a third year student of agriculture. So sir, my question is, যখন আমরা ট্রান্সজেনিক প্লান্ট যখন আমরা চাষ করি তখন সেটাকে যখন আমরা মানে আশেপাশের ফিল্ডে যাতে সেটা কন্টামিনেশন না হয় বা সেটার থেকে পোলেন গিয়ে যদি আশেপাশের নন ট্রান্সজেনিক প্লান্টকে যাতে ফার্টিলাইজ না করে, সেটার জন্য আমরা অনেক মেনটেন করার চেষ্টা করি বা ফার্মারকে অনেক মেনটেন করতে হয়, বাট সেটা তো 100% মেনটেন হয়ে ওঠে না বা সেটাকে সম্পূর্ণভাবে তো কখনো মেনটেন করা যায় না যাতে মানে কন্টামিনেশনটা না হয়। তো এটার জন্য কি উপায় আছে?
না, এটার জন্য কোনো উপায় নেই।
রিফিউজি চাষ করতে বলা হয়, এটার জন্য কোনো উপায় নেই। তবে আমরা অনেক ট্রান্সজেনিক প্লান্ট চাষ করছি, তোমায় কে বললো? আমরা একটাই ট্রান্সজেনিক প্লান্ট চাষ করি, দ্যাট ইজ বিটি কটন। আর কোনো ট্রান্সজেনিক, শোনো, এইসব গপ্প টপ্প যারা করেন তারা এটা গপ্প করেন, তারা কোনো হচ্ছে পিয়ার রিভিউয়ের লোক নয়, গপ্প করে যান। আমরা অনেক ট্রান্সজেনিক প্ল্যান্ট করছি না, আমরা অনেক ট্রান্সজেনিক কিছু করছি না, এইটা হচ্ছে ইয়ে। এইবারে তোমাকে একটা প্রশ্ন করি, তো পোলেন দিয়ে কি যাবে? না না, পোলেন দিয়ে গ্যামেট যাবে তো, সে তো ঠিক কথা আছে। গ্যামেট মানে তোমার একটা ইয়ে যাবে, একটা হচ্ছে পোলেন গ্রেন যাবে, তার মধ্যে দিয়ে স্পার্মটা চলে যাবে। না কি জিন যেতে, তুমি তো জিন টিনের কথা বলবে না, কি জিন তুমি ভাবছো যাবে, মানে যাতে তোমার ক্ষতি হবে? না, আমি তোমায় কি বললাম, genes are not meant for one organization. তোমার মধ্যে, তোমার শরীরে একটা জিন আছে, যে জিনটা তোমার হচ্ছে চুলটা কালো হচ্ছে। ইউরোপিয়ানদের চুলটা কালো হয় না। এই জিনটাকে যদি আমি সরিয়ে নিয়ে গিয়ে আমি যদি অন্য একটা হচ্ছে মানে হচ্ছে যে অ্যানিমেলের মধ্যে দিতে পারি, যদি সে নেয়, তাহলে কিন্তু তার লোমটা কালোই হবে। তাহলে তাহলে কি ক্ষতি হবে তার? এই জিনটা যে প্রোটিন, জিন মানে এটা একটা প্রোটিন তৈরি করবে। সেই প্রোটিনটা যদি তোমার শরীরের পক্ষে ডেট্রিমেন্টাল না হয়, তাহলে তোমার ক্ষতিটা কি? ক্ষতিটা তো নয়। এইবারে ধরো তোমাকে একটা কথা বলি, আমি তোমাকে যে বললাম যে ব্যাসিলাস থুরিজেন্সিস থেকে দুটো জিন তুলে নিয়ে গিয়ে বিটি কটন তৈরি করেছে। কি জিন তুলেছে? না ইনসেকটিসাইডাল প্রোটিন তুলেছে। সেটা কি করে? না এটা ইনসেক্টকে কিল করে। হ্যাঁ, আচ্ছা। বিটি পাউডার নাম শুনেছো? আচ্ছা, ডক্টর ঘোষ আপনি তো জানেন বিটি পাউডার। তো বিটি পাউডার কিন্তু আমরা স্প্রে করতে বলি, মানে বিটি জিনটা মানে হচ্ছে বিটি পাউডার মানে হচ্ছে ব্যাসিলাস থুরিজেন্সিসে, বিটি মানে ব্যাসিলাস থুরিজেন্সিস, একটা সয়েল ব্যাকটেরিয়া। এইটার পাউডার, এই পাউডারটা আমরা যদি স্প্রে করি তাহলেও একই কাজ করে, মানে ওই ইনসেক্টটাকে কিল করে। তা পাউডারটা যখন স্প্রে করি তার মধ্যেও তো ওই ইনসেকটিসাইডাল প্রোটিনটা থাকে বলেই কিল করে। তো কিছুকে কিল করতে গেলে তার মধ্যে তো একটা কিছু একটা হচ্ছে যে সাইডাল কিছু থাকবে, মানে কিল করার ক্ষমতা থাকবে, তো একটা কিছু পয়জন তো থাকবে। সেই পয়জনটা যদি অনলি ফর দ্যাট ইনসেক্ট হয় আর তোমার জন্য না হয়, তাহলে তোমার ক্ষতি হবে কি? তাহলে কেন ক্ষতি হবে? তবে যদি পোলেন ট্রান্সফার তুমি না চাও, ইফ ইউ ডু নট, তাহলে তোমাকে রিফিউজি প্ল্যান্ট রাখতে হবে, কিন্তু বলো তো বিটি কটন চাষ কবে শুরু হয়েছে জানো? আজকে প্রায় তোমার 20 বছর হয়ে গেছে। তুমি শুনেছো বিটি কটনে কোনো মানুষের ক্যান্সার হয়েছে? কোনো রিপোর্ট, কোনো কাগজ, কোনো ইয়ে? পরিবেশবিদরা ওই সুমন সহায় বা তোমার ইয়ে কি বলছেন, ওনাদের কথা বাদ দাও, ওনারা অনেক দিন আগেই বলে গেছেন ক্যান্সার হচ্ছে। তুমি শুনেছো কোনো ডাক্তার, কোনো ইয়ে শুনেছো? তাহলে তুমি বলবে যে ওটাতে তো কাপড় হয়। তুমি জানো বিটি কটনের যে কটনের সিড থেকে তেল হয়, সেটা আমরা খাই, জানো না? কটন অয়েল হয় জানো না? সারা বিশ্বের হচ্ছে যে একটা হচ্ছে তেলের একটা বড় সোর্স হচ্ছে এডিবল অয়েলের একটা বড় সোর্স হচ্ছে কটন অয়েল। কারোর কিছু হয়েছে? তাহলে ট্রান্সজেনিকের জন্য যত কিছু মানে যত রকম মানে হচ্ছে সেফটি মেজার আছে, আমাদের নর্মাল ভ্যারাইটি তৈরি করার জন্য তত সেফটি মেজার কিন্তু মধ্যে দিয়ে যেতে হয় না। একটা ট্রান্সজেনিক ভ্যারাইটিকে পেতে গেলে যত রকম সেফটি মেজার দিয়ে যেতে হয়, সে ডক্টরস থেকে শুরু করে এনভায়রনমেন্টালিস্ট থেকে শুরু করে যত মানে এনভায়রনমেন্টাল মানে সায়েন্টিস্ট, নন সায়েন্টিফিক পরিবেশবিদ নয়, সায়েন্টিস্ট, তাদের মধ্যে দিয়ে যেতে হয়। আর কোনোটার মধ্যে অত যেতে হয় না। এবার তুমি বলো গমের মধ্যে কি আছে? হ্যাঁ, তাহলে এই যে গমের ভ্যারাইটি যে 16-64 করে আমরা তো চেল্লালাম, ওটাও তো ক্ষতিকর। ওটার মধ্যে তো গ্লুটেন আছে। তুমি গ্লুটেনে তোমার কিছু হয় না, কিন্তু যার হয় সাংঘাতিক হয়। তাহলে চিংড়ি মাছ খাও, আবার দেখো অনেকে খায় না। তাহলে অ্যালার্জি তো একটা প্রোটিন, তো তোমার হতে পারে এইটাতে, আমার হয় না। তো আগেও ছিল, এখনো আছে, বাদামে অনেকের হয়। তো তো এগুলো হচ্ছে না। ট্রান্সজেনিক আমরা অনেক চাষ করি না, 1 নম্বর তোমার প্রশ্নের উত্তর। নাম্বার 2 হচ্ছে হ্যাঁ, রিফিউজ করা উচিত। যদি তুমি পোলেন বন্ধ, আর যদি তুমি মনে করো যে পোলেনের মধ্যে দিয়ে যদি ধরো তোমার ওই জিনটা চলে যায়, তো তোমার কি অসুবিধা? তোমার তো ভালো, তো তোমার তো ভালো। আর তাছাড়া ওইটা তোমার জানা কথা যে ওইটা হচ্ছে মানে যাদের অ্যালকালাইন গাট,
Okay, thanks again to Dr. Subhendudev Chatterjee for his nice discussion and interaction with the student. And you know that is a part of academic activities of our School of Agricultural Sciences, and it is in our academic calendar. So we have 2 genetics faculty, Dr. Umme Salma and Dr. Polomi Sen. They will take you follow-up action in your classroom, and they will discuss further if you need. So all the pivotal activity and this to organize this, I shall thank to Dr. Umme Salma, and I shall make over the mic to him, to her, to next of the program or any vote of thanks or like that. So I thanks all. I thanks to the speaker and the audience and our faculty, those who are instrumental to make the program very success and grand success. Furthermore, we shall carry out another program in another time by the organized School of Agricultural Science in this academic area. But we are committed to do it. That's why according to academic commitment we are doing it. So it has got great interpretation, it has got great implication, and it has got great influence in your academic preparation throughout the journey from first year to fourth year. Now we have the first fourth year student, they will be benefited more because they will take the master degree, PhD in the specific subject. So many students can come, so they will do the advanced research. There is no end of research, it is going on from the 1947 to till now. You have heard the history, how the food production of India increased gradually, and in what point we have reached, but our population also is increasing day by day. We have now 140 million, 140 crore, 140 crore of people in our country. So we have to more and more emphasis we have to give into food production, but we have to take the precaution so that environment cannot be distorted, the equilibrium cannot be distorted. We cannot put in the further disastrous situation like cyclone, like so many natural disasters that we have to face. We have to check it, and we have to go ahead and go advance. Okay. Thank you very much. Thank you again, because I am giving the, I am give over the mic to Dr. Umesh Sharma for next part of the program.
Thank you. Thank you.
Thank you, sir. It is my humble request to the guest and our director, sir. Please, all of the faculty come on the stage for a photograph, official photograph. And guest.